Hello students, today in this video we're going to discuss about the process of transcription. Now what is transcription? First of all, transcription means the process of carrying your genetic information from one strand of DNA to RNA. Remember that it is only one strand. Since DNA have double-stranded, but in the process of transcription, only one strand of DNA will be used for the process, for the synthesis of RNA. So this is your DNA to RNA. So it will be only one strand. We'll discuss why only one strand. Why don't we use both the strands, okay? Now, first of all, we have to understand heterocatalytic. Heterocatalytic. The meaning of heterocatalytic means DNA will be helping for the synthesis of RNA and for the synthesis of protein. So that is said to be heterocatalytic. Okay. I'm repeating again. Heterocatalytic means DNA will be helping for the synthesis of RNA and for the synthesis of protein. So for the synthesis of DNA to RNA, we call what? Transcription. For the RNA to protein, we call translation. The next important thing is about DNA template. We need a DNA template for the process of transcription. Okay. This is 3' to 5', and here we have 5' to 3'. Okay, 5' to 3' is considered as coding strand. Coding strand. Now, what is coding strand? Now, in this strand, there will be nitrogenous bases which is going to code for amino acids. You have to know that proteins are made up of amino acids. Okay, just say that one amino acids, second amino acid, third amino acid. As many amino acids, if they keep on joining, protein is formed. But to code for these amino acids, like which amino acid will come first, okay, depends on the code. There is a code that is called genetic code, and usually that is of 3 codon. Okay, means 3 nitrogenous base will give one code. For example, we have A, U, G, C. Just say A, T, G, C and A, T. So these 3 nitrogenous base will code for one amino acids, and these 3 nitrogenous base will code for another amino acids. Now, this is not used as a DNA template. We use DNA template as 3' to 5'. Why? Okay. If you see the opposite strand, anti-parallel strand, then A will be having T, right? And T will be having A, C will be having, G will be having C, then this is G, A will be having T, T will be A. Now, if from this strand, just assume that this strand is going to give RNA. Okay, this is your DNA strand that is going to give RNA. Now, for this strand I am making here. So A will be, for RNA, A changed to U, uracil, right? So T will be A, G will be C, C will be G, then A will be U, again T will be A. Okay. For this, if you see this, T will be A will be U, C will be G, C, then T will be A, then A will be U. Okay. So if you see this code A, U, G, C, A and U. So you will find in the case of 3' to 5'. Okay. So this template strand will provide, the coding sequences, the coding sequences that is there in the 5 prime to 3 prime. But if you use this as a template strand, if you use this as a template strand 5 prime to 3 prime, then you get this U A C G U A. So see, you can see that this sequence is not matched with this, right? Assume that this is your mRNA, this is your RNA, which is synthesized in the process of transcription. If you use this strand, then you get this UAC. So it is different, right? All the sequences are different. See, U, A is U, then it is A again, this is G is equal to C again. So this nitrogenous base is completely not matched with the coding strand. But if you use this is matched with this coding strand. Okay, so we have to use DNA template as 3 prime to 5 prime. Okay, always remember that we have to use DNA template as 3 prime to 5 prime for the synthesis of RNA. Now the next important thing is about third is enzymes. Enzymes. So enzymes are needed for the process of transcription. So the first enzyme is DNA polymerase. DNA polymerase. Not DNA, it's RNA polymerase. Okay. Now RNA polymerase for both prokaryotes. For prokaryotes, it is only one RNA polymerase. Okay, and for prokaryotes, we have only one type of RNA polymerase, and they can synthesize. This will synthesize your rRNA, means mRNA, and tRNA. All the three different types of RNA can be synthesized by RNA polymerase. For eukaryotes, there are three types of RNA polymerase. Okay, three types. That is RNA polymerase 1, 2, and 3. There are three types of RNA polymerase. RNA polymerase 1 will synthesize rRNA. Okay, and in rRNA there are again subunits. Okay, there is 28 A's. Okay, we have 18 A's, and then we have 5.8 A's. So what is S? S means sedimentation coefficient. Okay. Now in RNA polymerase 2, we have hnRNA plus mRNA. So what is hnRNA? hnRNA is heteronuclear RNA, and we have the third type, that is RNA polymerase 3. It's going to give your tRNA and small nuclear RNA. Okay, so these are the three different types of RNA polymerase that you find in eukaryotes. So this enzyme is very, very important because its activity is always 5 prime to 3 prime directions. Okay, its activity is 5 prime to 3 prime directions. Let's discuss the structure of the RNA polymerase. Consists of 7 subunits. Okay, and out of that, the 5 subunits are, we have 2 alpha, okay, and we have 1 beta dashed, 1 beta, and we have 1 omega. Okay, so 2, 3, 4, 5, and the another one is just the it's the subunits, but these are needed as requirement. Okay, whenever it's required, these subunits will come to an action. That is sigma factor. Okay, I'll just write in spelling. Sigma factor. And the another one is rho factor. So we have 2 alpha, okay, which is going to bind to the initiator protein. Binds to the initiator protein. This alpha, this is beta dashed, okay. This will be binding, this will bind to the DNA template. DNA template. Okay. This will bind to your nucleotides. Sigma factor usually help for the initiation process, okay. Initiation process. We're going to discuss in detail about that. Then we have Rho factor. This is elongation, will help in the process of elongation and termination. So RNA polymerase have 7 subunits. 5 subunits are very important. Okay, we have 2 alpha, we have 1 beta dash, 1 beta, and 1 omega. 2 subunits. Depending on the requirement, they come to an actions and they leave the RNA polymerase. Sigma factor, which will be helping for the initiation of your RNA synthesis, and Rho factor, which is for the elongation and termination. Before understanding the process of transcription, we have to understand the structure or the area where this transcription occurs. And to understand that, we have to know the structure of transcriptional unit. So this is our transcriptional unit. Just say that this is 3' to 5', and this is 5' to 3'. So this is double stranded. Now this is your, you know that 5' to 3' is your coding strand, and this is your template strand. We're discussing on transcriptional unit. Now, here in this position, okay, this is said to be the promoter. Okay, and this area is said to be the terminator. Okay. Now, what does promoter means? In promoter, it is a point or it is a position from where the transcription is going to start. Okay, so this will be the area where the transcription will start. Now, promoter usually have a sequences called TATA. Okay, TATA. What does it mean? It's said to be TATA boxes. Okay, TATA boxes. Now in prokaryotes, we call as, Prigno, P-R-I-B-N-O-W, Prigno boxes, okay? And in eukaryotes, we call as Goldberg, Hockness boxes. So this promoter has its name called as, in prokaryotes we call Prigno boxes, and in eukaryotes we call Goldberg Hockness boxes. Likewise, if I have to explain, okay, this third stream, we say to be 2 type of streams are named. In the 3' we call as downstream, okay. And the 5' is said to be the upstream. Okay. We have 3' as downstream and the 5' as the upstream. Likewise, the 5' is always upstream and the 3' is always downstream. So in MCQ, these questions can be asked. Said that in a transcriptional unit, the promoter is at the 5th carbon, that is the upstream of the coding strand. And we can also say that the promoter is at the 3', that is 3 carbon, that is in the downstream of the template strand. Likewise, for the terminator, you can say that the terminator is at the 5', that is in the upstream of the template strand, and downstream at the, that is 3' of the coding strand. So this is about transcriptional unit. This another important structure is said to be structural gene. Structural gene. So this is responsible as the unit of inheritance. Okay, this is said to be the unit of inheritance. It means that this promoter and terminator sequences, okay, for example TA, TA, these sequences are not going to help for the synthesis of protein. Okay, only the structural gene will be responsible for the formation of useful protein. Okay, so from this area the transcription is going to start. Okay, and from this area for coding to code the amino acids. Okay, from this area, just say that from this position to this position, okay, the mRNA will start synthesizing. Okay, so in the next class we are going to discuss about the detailed process of transcription and the RNA modification or RNA processing.